Namaskar and welcome to CEC Gurukul Lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. In this series of lectures on various analytical techniques applied in plant sciences, we have come a long way, and till this time we have covered various principles of microscopy. Going further, we have also covered different types of microscopes, light microscopes and electron microscopes. In the previous lecture, we have understood and discussed the basic principles behind electron microscopy, the basic concepts of resolution, and the various types of electron microscopy, and especially the transmission electron microscopes. Today we are going to discuss another very important variant of electron microscope, that is also termed as scanning electron microscopes. Electron microscopes, they work on certain principles and they have certain variations in terms of their application and also in terms of their principles. In today's lecture, we are going to discuss various aspects of scanning electron microscopes. So let's talk about the history of scanning electron microscope. Scanning electron microscope was invented by Manfred von Ardenne in 1937. In scanning electron microscope, the image of the specimen is produced with a focused electron beam that is scanned across the area of the specimen. In scanning electron microscope, now we shall be calling it SEM. So in SEM, a magnetic lens system focuses the beam of electrons into an intense spot on the surface of the specimen. So let us try to understand the basic structure of scanning electron microscope with the help of a schematic diagram. So in this diagram, you can very well see that in a scanning electron microscope, the electron beams bombarded or they are emitted with the help of an electron gun, and these electron beams, they are focused with the help of magnetic lenses, and these focused beam of electrons, they are projected on the samples and the surface of the sample based on its own contortions or surface topography, and these deflected electrons, they are perceived by a detector. So in case of scanning electron microscope, the samples are bombarded with the beam of electrons on its surface, and the electrons are then deflected back. In contrast to transmission electron microscope, where the focused beam of electron passes through the sample or specimen and their diversion or their refraction is detected by a photographic plate. In case of scanning electron microscope, the spot is moved back and forth across the specimen by charged plates called as beam deflectors. These beam deflectors, they are located between the condenser lens and the specimen. The beam deflectors attract or repel the beam according to signals sent by the deflector circuitry. As the electron beam sweeps rapidly over the specimen, molecules in the specimen get excited to higher energy level and they start emitting secondary electrons which are then used to form an image of specimen surface. We can understand this entire process with the help of this schematic diagram, where we can see that the electron gun emits the beam of electrons, and the condenser lens further focuses these electrons in the form of a beam, and then it passes through the objective lens before it hits the specimen. In between condenser lens and the objective lens, there lies a beam deflector, and the electrons which are deflected from the specimen surface, they are detected by a sensitive receptor called as detector, and then a scan is generated while comparing the beam of electrons from the beam deflector and the detector sensors, and then it is computerized into a picture on a monitor. So the secondary electrons are captured by detector located immediately above and to one side of the specimen. The essential component of the detector is scintillator. Scintillator, when excited by electrons incident upon it, it emits photons of light, and these photons are used to generate an electronic signal onto the video screen. So this is the detector, which has the scintillator converts this secondary electron messages into photons, and these photons are used to create an image on the monitor. So as the beam traverses the surface of the object, electrons are deflected to various degrees. The deflected and emitted electrons are detected by a photo multiplier tube and used to form a three-dimensional image of the object's surface features. As far as the resolving power of scanning electron microscope is concerned, it is obviously very less in comparison to the resolving power of transmission electron microscope. However, since the image formation by scanning electron microscope is dependent on surface properties, it can magnify samples up to many centimeters, and thus it has a greater depth of field also. It can thus produce good representative images of the three-dimensional shape of the sample. So if we have to compare the resolving power of various optical systems, so the resolving power of an unaided eye is merely 0.1 millimeter, whereas a good light microscope can have a resolving power of up to 0.2 micrometer. When we compare this resolving power with scanning electron microscope, the scanning electron microscope has a resolution of 1 nanometer, whereas the resolution power or resolving power of transmission electron microscope is as fine as 0.2 nanometer. So this is the basic diagram which explains the entire functioning of scanning electron microscope. The electron beams are emitted from the electron gun because of the voltage difference between the cathode and the anode ring. So higher the voltage difference, the higher will be the intensity of electron beams. These electron beams, they pass through magnetic lenses generally made up of copper and other transition elements. These magnetic lenses, they focus these electron beam towards the sample, and before it reaches to the sample, there are other scanning coils and other types of lenses which further intensify these electron beams. And these scanning coils and other lenses, they are also attached to a TV scanner. So when the beam of electron falls on the specimen, they deflect these electrons in various degrees depending upon their surface topography. They are these reflected or deflected electrons are called as secondary electrons, and these secondary electrons are perceived by secondary electron detectors. So there is a direct comparison between non-deflected or the primary electron beams and the secondary electrons, and this creates a computer generated image so that the minute or the finer details of the sample topography could be observed. So the second diagram also tells us that this incident beam which falls on the surface, it generates different types of rays. One is backscattered electrons, the other one is secondary electrons. It also generates some amount of X-rays and also generates Auger electrons. So all of these reflected electron and various secondary types of electrons, they are used to perceive a computer generated image of the sample surface. Now, after understanding the basic principle of scanning electron microscope, let us try and understand how the samples are prepared for observation under scanning electron microscope. So first, the material is primarily fixed by immersing in 2.8 or 3% of glutaraldehyde in 0.1 molar Hipis buffer at pH 7.2 with 0.02% Triton X100. This process is done for several hours at room temperature or overnight at 4 degrees Celsius. This treatment of any material with glutaraldehyde fixes all and binds all the proteins at its own position, so that while further preparing the sample, these proteins and other components, they do not move their places. After this overnight treatment, the material is then washed multiple times in 0.1 molar Hipis buffer at pH 7.2. Now, after the washing of glutaraldehyde with the buffer, the next step is of dehydration. So dehydration process is done for different time periods at different concentrations of alcohol. So it starts with 10 minutes treatment in 25% of ethanol, followed by 10 minutes in 50% of ethanol, 10% in 70% of ethanol, and then 10% in 85% of ethanol. As we proceed, the concentration of ethanol gradually increases. So it is further treated for 10 minutes in 95% ethanol, and twice it is treated for 10 minutes in 100% ethanol. The ethanol finally used happens to be of EM grade. Now this dehydration process is followed by critical point drying. This process is an automated process which takes up to 40 minutes to get completed. After the critical point drying process, the sample is mounted onto a metal stub with double-sided carbon tape. And then finally, a sputter coating is done by applying a thin layer of metals. These metals could be of gold or palladium. This sputter coating is done over the sample using an automated sputter coater. So this entire process of preparation of any sample can be summarized with the help of this diagram. Any sample from any biological object, they are first treated in glutaraldehyde. They are fixed and they are fixed overnight, and then after the process of fixation of any sample, it is then transferred for the process of dehydration. Dehydration takes place through a series of alcohol concentration ranging from 25% alcohol to 30%, 50%, and reaching up to 100% alcohol for 10 minutes each. Now, once the dehydration process is done, the sample is processed or sample is sent for critical point drying for 40 minutes to 2 hours. Now, on the sample is covered with carbon sheet on both the sides, and on this carbon sheet metal coating is done with the help of sputter coater. These metal coating is done by applying gold or palladium metal. This coating is so thin that it exactly conforms to the surface contortion or the surface topography of the sample. In this process, the organic sample is gradually washed out, and the metal coating which conforms to the contortions or the topography of the surface remains there, and then this sample is mounted on a metal stub. It is mounted on a disc of metal stub for further scanning electron microscope observation. So the entire process can take up to 80 to 100 hours. So now let us talk about some of the variations of scanning electron microscope. One of the foremost variation or the modification of scanning electron microscope is scanning transmission electron microscope, also called STEM in short form. So what happens in STEM? STEM contains elements of both transmission electron microscope as well as scanning electron microscope. Like scanning electron microscope, it uses an electron beam that sweeps over the specimen. The image is formed by the electrons transmitted through the specimen as with transmission electron microscope. The STEM or the scanning transmission electron microscope is capable of distinguishing specific characteristic of electron that are transmitted by the specimen, thus deriving information about the specimen not obtainable with the conventional transmission electron microscope. So this is a diagrammatic representation of scanning transmission electron microscope, where electron beams are generated from the electron gun. Then it passes through a system of condenser lenses, then condenser apertures. They are focused with a deflection scan coil, and then it passes through upper objective pole piece, and then these beams, they pass through the specimen. and these transmitted electron beams and the deflected electron beams, because of the internal structure of the specimen, it passes through the lower objective pole piece, and then at the end these electron beams, they are received by STEM detector, and these STEM detector, they create an image which is much more finer in detail compared to the scanning electron microscope, and it also gives us a very good idea about the internal structures of the specimen. The other type of variation of scanning electron microscope is called as environmental scanning electron microscope, also termed as ESEM. So ESEM is a specialized scanning electron microscope which makes possible to perform microanalysis on uncoated specimens. So this is a very major breakthrough where the specimen need not necessarily be coated with metals, and it can also helps us in performing various degrees of microanalysis. Environmental scanning electron microscope, they also allow the specimen to be observed without freezing, without coating, without fixing, and without embedding. So this type of scanning electron microscope uses special technique, or I would say they uses a higher degree of vacuum and other modifications, so that the requirement of freezing or coating or fixing or embedding of the material is not mandatory. So here we can observe, we can see an environmental scanning electron microscope, a schematic diagram where electron passes through the anode aperture and then followed by column lining. It passes through the projection aperture, then the column isolation valve, upper pressure is very high, and then the vacuum is maintained manifold times, and then the electron passes through the specimen. The electron which passes or which falls on the sample, it is received by the detector electrodes. So that was our environmental scanning electron microscope. So in our next lecture, we shall try to understand the various other variations and various other methods of preparation of sample apart from the sectioning, and we shall also be able to understand that what are basic requirements when we cut a section through microtome, and then we place them for observation under scanning electron microscope. Thank you so much for your patient listening.